So welcome to one of the parallel sessions, which is going to be focused on materials and microbial systems. And for those of you who weren't here earlier, I'm Nick Abbott and have the task of coordinating this technical session. And really the purpose of the session is to highlight some of the research which is going on at the university. And what I wanted to do is to just preface the three presentations that we have this morning with a few comments. The first is just to remind you of the importance of microbial systems. And microbial systems come up in all sorts of contexts, and probably when we first think about microbial systems, we think about negative things like pathogenic organisms in food or wound infections, and but of course there are many good things that come from the presence of microbial systems also. And that goes back also to things like foods, and so processes such as pickling, for example, bioremediation and treatment of sewage. I mean, these are very important things and involve the engineering of microbial systems. And then, more recently, although actually also with a very long history, the idea of taking advantage of microbial systems as chemical reactors for making a range of chemical species which are actually very, very difficult to make through conventional synthetic pathways. So, there's a very compelling reason to explore microbial systems, and of course, in the context of this industrial consortium, the thing that I wanted to note is that to make progress in many of these areas, we need to understand how to design material systems that interact with microbial systems in useful ways. And so that's sort of what you're going to hear about this morning. And one can ask the question, well, to make progress in this area, what do we need to know? And so I would argue that two sort of fundamental challenges that we need to address are these. And the first is to understand more about bacteria. And unlike eukaryotic organisms, we know in many cases remarkably little about bacteria and how, for example, bacteria assemble themselves. And if we do understand a bit more about, the design principles that bacteria have evolved to adopt, I think it'll point us towards rational approaches to engineering interactions with them. And then the second challenge is to understand better how chemical and physical environments influence bacterial behaviors. And that's also something that I find amazing, is that we don't really know how, for example, complex rheological environments affect bacterial behaviors. And so these two sets of questions, you'll see efforts to address them embedded in the three presentations that you hear this morning. And before I introduce the first speaker, I wanted just to highlight two other aspects of efforts which are ongoing at the university, which do touch on these questions. And they are efforts which have been pursued within the MRSEC.
If I can interrupt, could you move your mic up? It'll make for a better recording. Okay, perfect.
And so the first comes this question about how bacteria assemble themselves. And this is really a focus of research in Professor Doug Weibel's laboratory in the biochemistry department here at the UW. And so what's depicted on the left here is a representation of a bacterial cell, and it's capable of amazingly complex functions. And one can ask the question, you know, how is it that bacterial cells control their organization? And unlike a eukaryotic cell, they don't have a cytoskeleton to direct internal components. But when you start to label certain molecules within bacterial cells, they're not randomly located, but they have well-defined organizations. And so one example of this is shown on the right side, and again, this is work from Doug Weibel's laboratory. And what they did here was they stained bacterial cells with a dye, and a dye that associates with a particular lipid found in the membrane of the bacterial cell, and the lipid's called cardiolipin. And what you find is that the dye doesn't uniformly stain the surface of the cell, but it's localized. And so it's telling you, or it's suggesting, that this lipid is distributed non-uniformly through the cell membrane. And so again, the question is, well, how does the cell do that? Because it doesn't have this internal skeleton to direct the organization. And as Doug has sort of documented, in fact, this sort of polar localization of proteins is also very common in bacterial cells. And so, you know, it's an intriguing question. And so how is the organism achieving this localization or positioning of specific components? And all of these different proteins, which are being labeled in these micrographs, carry out particular functions which are important to the cell survival. And so the answer is actually very simple. And the answer is that bacteria are great strain engineers. So we hear a lot about, you know, engineering of strain in synthetic material systems, thin films of semiconductors to control defects. Well, bacteria have been doing it for a long time. And so what Doug did, he did this beautiful experiment to make this point. And what he did was he took the outer membranes of the bacterial cell and he forced them against curved surfaces. And so what he did was he used microfabrication to create little microwells. And you'll notice that each of these microwells has a different curvature. And then he used osmotic pressure differences across the membrane to force the membranes to adopt the curvature of these microwells. And so what's plotted here is the curvature of the microwell, and this is telling you about the location of that particular lipid, cardiolipin, present within that bacterial membrane. And what you see is that if the curvature is low, the lipid is relatively uniformly distributed across the membrane. But if the curvature is high, then you start to see the lipid localize. And indeed, the curvature of the microwell is similar to the curvature of the end of the cell when you start to see the localization. And so what is the cell doing? So the cell is making lipids, lipids that have particular shapes. And so by placing them into curved environments, you can minimize the elastic strain in that cellular membrane. And that can be formalized using sort of continuum descriptions of the elastic properties of membranes. And so you know, I think it's a very interesting sort of observation. And in fact, it's one that we have taken to heart in a variety of synthetic systems now, showing that we can engineer elastic energy in synthetic membranes to achieve localization of non-biological species. But if you understand that this is how cells are regulating the positions of components of membranes, it also hints at ways of controlling the interaction with the bacterial cell. And so the second question that I mentioned, and I'd wanted just to highlight one thrust of research which addresses it, is the question of how the chemical and physical environment of cells impacts their behavior. And if you think about bacterial systems, where bacteria live, they live in very complicated environments. So they don't live in PBS or in simple cell culture media that you might typically do an experiment with in a laboratory. But they live in very complicated biological fluids. They live in mucous membranes, for example, that have very complicated rheological properties. And in some cases, it's thought that the environments are anisotropic. Anisotropic, for example, due to the presence of shear. And so one can ask a very simple question, and that is, how does anisotropy in the environment of a motile bacterial cell impact fundamental cell behaviors? And the answer is, it has a profound influence. So how can one address that question? And so we go to the world of material science, and we use liquid crystals to create anisotropic environments for cells, bacterial cells. And so there's a class of liquid crystals that are water-based and are formed from these molecules. So it's called disodium chromoglycate. It was a molecule actually first developed as a drug for asthma, and it turns out that if you add it at relatively high concentrations, 10 or 15 weight% to an aqueous solution, it will assemble into these nanoscopic fibrils, which will then organize to have long-range orientational order, which is sort of the definition of a liquid crystal. And so it's the same sort of stuff that's in a liquid crystal display, but it's water-based rather than being purely organic. And so this is just the birefringent texture of the liquid crystal, and we're heating it up, and ultimately it will turn into an isotropic fluid. And so these materials have anisotropy. So they have long-range orientational order of these little nanoscopic fibrils. And so the question is, how does that anisotropy influence bacterial behaviors? And so Doug Weibel introduced us to this organism called Proteus mirabilis. And it's a rod-shaped gram-negative bacterium. And it's a so-called pusher. So it has a flagella that rotates. And this is a typical image of the bacteria buzzing around in an isotropic fluid. So this is not the liquid crystal, but this is an isotropic environment. And the characteristic motion of the cells is what's called sort of a run and tumble sequence. And so what happens is it rotates its flagella, pushes it through the fluid, and at some point in time it reverses the direction of rotation of the flagella, and that causes the organism to tumble, and then it starts off on another run. And that process gives it an opportunity to change direction and couple, for example, to the chemical environment. So it's a very sort of clever way to migrate. And so the question is, what does an anisotropic environment do to these cells? And so on the right is again the cells in an isotropic cell culture media, and so it's undergoing this sort of run and tumble mechanism. On the left are the cells in the liquid crystal. And the liquid crystal is aligned so that these little fibrils are pointing horizontally. And so what you notice is that the cells have fundamentally changed their behavior, and they are being guided along by that anisotropic viscoelastic environment of the liquid crystal. And so the liquid crystal sort of behaves as a rail, and the bacteria just move along it. And not only does it have this sort of profound impact on the bacterial motility, but it does a bunch of other things to the cells. For example, it leads to additional mechanisms by which cells can interact with each other. So the liquid crystal is an elastic medium, and you can have the overlap of strain fields between the cells, and that leads to intercellular interactions. So if you notice, these two cells come towards each other, they find each other, and they go off together. And they're going off together, held together by the elasticity of the liquid crystal. And so it hints at ways of creating new types of interactions, so potentially sort of communal behaviors of bacteria. And then finally, we can sort of start to put on our engineering hats and ask, well, what might we do with this phenomena that could be potentially useful? And it turns out that you can create what are, in effect, bacterial diodes using the liquid crystal. And it's all based on the elasticity of the liquid crystal. And so the idea is that the cells will follow the orientation of the liquid crystal. So if we engineer into the liquid crystal particular patterns in orientation, we can use those to guide cellular behaviors. So the pattern that we're showing here is that we've created this distortion in the liquid crystal. This is a side view. And then we place bacterial cells into the system. And so if the cells are going from right to left, they will just follow the direction of the liquid crystal, which points them in the left direction. If they go from left to right, what happens is that they will turn left and they'll start to go up towards the vertical. So if they flip direction, they can't go very far left to right, but if they're going right to left, they can keep moving. And so if we just look at a movie, you see this P is indicating the direction of the strain and the liquid crystal. And so what you'll see is that cells can come in on here and they can move along, going to the left here. If they try and change direction, as this one will, and it tries to go back to the right, it's forced up into the vertical, so it takes on a black appearance when you're looking at the end of the cell. And so if you have a population of bacteria in this environment, you can localize the bacteria through the fact that they can move only to the left. And so one can think of ways of guiding a very dilute population of bacteria into a specific location in space. And so that sort of hints at analytical methods for finding bacteria in very dilute samples. And so that's something that we're very interested in pursuing. And it emerges out of this idea of just trying to understand, how does the environment influence the behavior of bacteria. Okay, so that's all I'm going to say as a preface to the three presentations which are going to follow. And again, just think about these presentations in the context of understanding how bacteria create themselves and how they interact with their environments, and they all map onto those two questions. And so the first one I like a lot. It's going to be about bacterial walls, and given the current presidential election, I think it's timely to think about how to build walls, of course, and so how do bacteria build walls, right? So fortunately it's not between countries. And then we'll talk about how the properties of surfaces can influence bacteria, particularly slippery surfaces. And then finally, we'll hear a presentation which will be about how to make surfaces more effective in killing bacteria. So that's going to be the sequence of the three presentations. And so the first one is going to be given by Rishi Trivedi from Professor Doug Weibel's laboratory. And so Rishi is a PhD student. He's part of the MRSEC, and he's also a co-founder of a student organization called Future Science Leaders. And so he has very broad interests, and he's also doing a minor in the business school. So many interests and is taking full advantage of the campus. And so Rishi, why don't you come up?